Okay, so this is a very simple drawing. So basically our cerebellum has got three parts. Okay, it's got the cerebellar cortex, it's got the inner white layer and another layer of deep cerebellar nuclei, which is the part of the gray matter only. So now if we were to take the cerebral cortex alone, it's got three layers. It's got the outer molecular layer, it's got the inner middle Purkinje layer and the inner granular layer. So these star-shaped neurons that you see over here are the stellate nuclei, and the basket are basket cell shaped nuclei. These are going to be Purkinje cells, and this is going to be the granule cells, and this is going to be the Golgi bodies. Okay, so the granule cells alone are going to be in the opposite direction. That is, their dendrites are going to face towards the cerebellar nuclei, whereas their axons are going to move up towards the over here you will be having the pia matter. Okay, so it's going to be moving up, whereas all the rest is going to be moving down, and this granule cell is the only excitatory cell. All the rest of it are going to be inhibitory cell. So the granule cell is the axon is going to go and it's going to split into T shape, and it is going to innervate all the rest of the cells. Okay, it is going to innervate most of the other cells. Okay, and this is going to receive efference from the deep cerebellar nuclei as well, and it is going to get the input neurons from the three dots you see over here. So this is representing the mossy fibers, which is going to arise from the pontine nuclei, from the vestibule and all, and from the inferior olivary nucleus you're going to have the climbing fibers. So the climbing fibers is going to directly go and it is going to affect the Purkinje fibers, which is going to inhibit the deep cerebellar nuclei, whereas this mossy fibers, it is going to go and stimulate the granule cells, which is then going to go and excite the other cells, which through feedback inhibition it is going to inhibit itself. Yes, so the granule cell is going to initiate this one. It's going to excite the Golgi cell, and the Golgi cell through feedback inhibition is going to inhibit the granule cell itself. That's how it is going to work everywhere else. Okay, whereas if we were to take a look at the feed forward inhibition also takes place over here, wherein one is going to inhibit the other and that is going to inhibit the other, and that can be seen when this granule cell is going to excite the Golgi cell, but the Golgi cell is going to inhibit the Purkinje cell. Yes, so this is feed forward, this is feedback inhibition. Okay, so this is the microcircuitry of the cerebellum.